All right. Good day to everybody that is joining us from across the world. We're really excited to be hosting this PolarConnect event. Today we have Cara and the research team. They are some there. I just noticed it says Arctic researchers on the title slide. They are Antarctic researchers. Sometimes we have issues with our polar regions here, but you are in the Antarctic and you're aboard the Nathaniel B. Palmer, a research vessel that we're going to hear more about. Today is October 3rd, 2016, and yes, very exciting moment for a lot of reasons because we can actually see, hear, and have a fantastic connection with Cara and the research team. So without further ado, I'm going to do a few slides before we turn over the presentation to the team. So the first slide is about the platform that we're using. It's called Adobe Connect. The slide should be changing in the center. You should see a list of participants. We have a webcam that is working today. We actually also have a file that you can download. It's an audio file that's going to be shown later on. There's a chat area, and if you haven't done so already, please type in your name and how many people are joining you so we get an idea of participants. And this event will be archived and we'll share it with everybody that registered as well as posted on the PolarTREC website at PolarTREC.com. You are already introducing yourself, so we'll skip on to why is Cara out there in the Southern Ocean with Bethany and the others that are working with them. It's because she's part of the PolarTREC program. It's funded by the National Science Foundation and administered by a nonprofit called the Arctic Research Consortium of the United States. We are primarily based in Fairbanks, Alaska, and have an office in Washington, D.C. PolarTREC is a program where we place teachers such as Cara with researchers such as Bethany, and they get hands-on field experience and learn all about science so that they can become better at teaching science in the classroom. And it's an exciting time of year for us as we have lots of teachers that are deploying to the field with scientists. We had 3 more that just went over the weekend towards Antarctica, so we'll be having several of these live events from the field coming from a variety of places around the continent of Antarctica. If you have questions as we go along, we ask that you type them in the chat for a little while, although I believe there is a couple of researchers standing by that can also answer questions in the chat as we go along while they present. And then at the end of the presentation, we'll remind you how, and if you want to ask your questions live to the team, we'll show you how to do that. So I am now going to turn this over to the team, and welcome. It's great to see you, ladies.
Thank you, Janet and Judy. First of all, just thanks to everybody for joining today. I have been overwhelmed by all of the wonderful comments and questions and the support of pretty much everyone that's been involved, and I'm having a wonderful time. So I hope my presentation today is going to tell you a little bit about what's going on the ship, although I do have to tell you there's a lot more going on than I have time to talk about today. So I'm going to give you kind of an overview of everything, and Bethany can certainly chime in if I'm saying something incorrectly or she wants to add something, and then I look forward to your questions at the end. So before I get into the science, I do want to take the time to introduce the science team. Dr. Bethany Jenkins, who's here with me today, is from the University of Rhode Island. She's a marine microbial biologist, and she has 3 graduate students here with her on the cruise. Dr. Drew Chappell is from Old Dominion University. She also has brought along 3 science compatriots with her. Kim Powell is her lab manager, and then she has 2 graduate students as well. And then from the University of South Florida, we have Dr. Kristen Buck. She is a chemical oceanographer. She specializes in trace metals, and she has a number of people with her. Travis, who, let me do this, see if I can do it, is right here. No, maybe not. little arrow. Travis joined us for the setup of the cruise and was not able to join us on the cruise, but he may join here shortly, so he'll be able to answer some questions on the chemistry side of things. We also have Dr. Randy Bundy with us here from the Woods Hole Oceanographic Institution. Excuse me. So a little bit about where we're actually studying okay, I'm going to try one more time to get the arrow. Oh, it's not popping up. You can see on the map on the left, the circular map. At the top is the southern portion of South America, where we actually flew into in Punta Arenas. And then across that big, vast ocean of blue is the Antarctic Peninsula. And the Antarctic Peninsula, we are focusing on the western side, but we are also sampling in some of the deeper waters there of that open ocean, which I believe, well, it's not labeled, but that is the infamous Drake Passage. So we've been sampling in the deep waters there, but also around the peninsula. And in the map on the right, you can see some of the sampling areas that we've actually been collecting water samples from. This area has a grid section known as the Palmer LTER, which is the long-term ecological research sampling area. And we've been using some of these sampling areas to collect water to learn more about the area and also in order to collect diatoms, which I'll talk about in a few minutes. This area has been sampled since 1990, so it's a wonderful area in order to not only add to the data that's being collected, but to be able to use data that was collected in the past in order to compare with what we're seeing here during this research cruise.
And I'd like to just add a quick point here. If you can see on the LTR grid map, we're following the 600 line currently, and all the lines south of there, we're having a little bit of an issue on our end with getting our pointer to work, but all the lines south of 600 in the lower numbers are currently encased in ice, and we will probably not be able to sample them.
Uh-oh, now I don't even have a slide. Okay, well, I'm not sure where that slide went or if you can see it, but the slide is supposed to introduce the ship that is taking us to all of these different locations, which is the research vessel and icebreaker Nathaniel B. Palmer. It was built in 1992, and it's owned by Edison Schwest Offshore. There are a number of crew members on board that keep the ship running for us while the science team can focus on the science. And there also is an additional set of folks from the Antarctic Support Contract, ASC, which are here to help support the science. So they run some of the science equipment, they help with the IT and with the electronic equipment, things of that nature. So there are actually a lot of people on board this ship, and it's a very large ship to be able to hold all of these people. It's 308 feet long. And it is one of very few U.S. icebreakers. So we're very fortunate that this ship, even though we can't get to some of the locations with thick ice, we are able to get to certain locations. There it is. There she is. We're able to get to certain locations because of the very thick hull. So that's the Palmer there on the top right-hand side of the slide. I have a picture here of the bridge, which is where the captain and the mates navigate and steer the vessel. And then on the lower right-hand side is a place where I spend a lot of time on the Palmer, which is the starboard A-frame, and I'll talk a little bit more about that in a moment when I talk about how we actually do our work here.
Yeah, and Cara,
the Palmer, yes.
Sorry. I just want you to know that we're caught up with the slides too, and did you stop your webcam?
Sure.
No, we lost your webcam. Oh, it stopped.
Oh, you did? Yeah. Okay. I did not stop the webcam, but I think I might for a moment then if we're having some problems. Maybe that's the issue. So let me shut that off.
Okay.
Okay. All right. Okay. So is it actually off?
I think our software on our end is having trouble responding. We can't even shut off the webcam. So if we have glitches, let us know.
Yeah. We'll figure it out as we go.
Okay.
Okay. The slide that you're seeing here is showing some of the rooms that are available for use on the ship. The top left-hand side is my cabin, which has 2 bunks and its own head or bathroom, so that's super convenient. The lower left-hand side is the dining area or a galley where we're served 3 meals a day, but the galley is open 24 hours a day in case you get a little craving later on in the evening. If we had 24-hour operations, we would also have a 4th meal served, but we're not doing that on this particular cruise. On the right-hand side of the screen are some of the laboratories that are available for scientists to use. There we go. All right, so we've lost our connectivity, so I'm just going to keep talking. And Janet, if you can flip the slides when I ask you to, that'd be great.
Yep, no problem.
Okay, so the labs, there are 5 main lab areas plus different cold rooms and other equipment rooms that we can use on board. These are available, and we're using most of them. And so I'll talk a little bit more about those labs as I go. The lab on the top right has a very weird-looking plastic bubble, and that bubble is the trace metal lab where some of the chemistry work is being done. And the lab on the bottom is the bio lab, and there are some microscopes and some other pieces of equipment there. You can go to the next slide, Janet. Thank you.
Okay.
So this is just showing you what it looks like to be underway in the Palmer. There are lots of days where we can have rough sea conditions, and there are some days when we experience ice like we're experiencing today. But the Palmer is built for this type of environment, and I will say that even on certain days, we had 25-foot seas, and honestly, it might have felt like it to some people, but not to me. So it's certainly a very stable vessel and able to cut through the ice very well. So that's a little bit about the ship that we're on and the location that we're sampling. And so I'd like to get a little bit into the science. So if you can go to the next slide. And I can't see if you're on the slides because we're, again, lost. So
You're good.
Okay. All right. So this is showing an Antarctic food web. And the food web is probably something that most people are familiar with. It shows how energy moves through an ecosystem. And most people are familiar with some of the animals that you'd see at the top of the food web. So animals like orcas or humpback whales and seals. But the focus of this research trip has to do with the organisms that you see in the red circle. So the sun, which is the source of energy for all organisms on Earth, somehow has to get its energy into that food web. And the organisms that are in the red circle are called phytoplankton. They are the primary producers that will take the energy from the sun and will convert it into food, putting that energy into the bonds of food, which is then passed through the food web. And this research team is working collaboratively to focus on a particular primary producer called a diatom. So Janet, if you could go to the next slide, please.
Okay.
These diatoms, these are some pictures that Bethany provided me of diatoms, and they're microscopic organisms. So we're talking about much smaller than even a millimeter in size. We're talking about micron units when we're talking about these diatoms. And diatoms are unicellular, and they're made of something called a silica frustule. So basically, they build themselves a little glass house, and that is a see-through surrounding that allows the sun to actually come through so that they can then produce that food through photosynthesis. There are between 14,000 and 18,000 different species of diatoms in the world, so there are a lot of different types of diatoms, and you can see that in the pictures with their various shapes, sizes, and even some projections that come out from the structures of these diatoms. I mentioned that diatoms photosynthesize, and so just so we're all on the same page, I'm not going to give you a chemistry lesson, but if you could go to the next slide, we're just going to look at a quick overview of photosynthesis.
Okay.
Photosynthesis, as most of my students hopefully know, and as some of you may know, is a chemical reaction that takes sunlight with water and carbon dioxide and ultimately produces food for these organisms as well as oxygen. And that's super important for us because we actually breathe in that oxygen in order to go through a process called cellular respiration. One thing that isn't commonly talked about is the center of the slide where I have the word nutrients. in addition to having sunlight, water, and carbon dioxide, there also are nutrients that are required for photosynthesis to be able to occur inside of these organisms. And one of those nutrients is iron, and this is a focus of the research that's going on this research cruise. Iron is the fourth most common metal on Earth. However, it's not necessarily that common in the oceans, and that is particularly true for the Southern Ocean. So here's where the research questions start to develop. If you can go to the next slide, please. Okay. So this, let me lay out what this picture is. On the left side of the screen, you'll see the label South America, and then if you look diagonally to the right, you'll see Antarctica there at the bottom. So those are showing the continental land masses, and all of the other sections of this diagram are showing you a three-dimensional image of the ocean, so some of the very deep areas and some of the very shallow areas. What's important to note on this diagram are the colors. So you'll notice that farther away from Antarctica, you can see colors like reds and yellows and even some of those peachy orange colors. That's an indication of higher iron concentrations in the ocean. If you look near the tip of Antarctica, the peninsula that we're actually doing our research near, you'll notice that most of the colors are blue and purple, and that's an indication of a very low amount of iron. So you can have a lot of sunlight during the summertime here, you can have a lot of carbon dioxide in the ocean, but if you don't have a lot of available iron, it might not be possible for these diatoms to actually survive, but they are surviving. So the question then is, how are they being successful here in an area with low iron? So there is a research objective to this entire cruise, and it's sort of a broad research objective that has a lot of components to it. So the next slide is going to have a lot of words on it, and they're going to be a little technical, but I'm going to break it down for you here in just a second. So next slide, please.
Okay. And this should be a slide entitled Research Objectives at the top.
Oh, okay. Sorry, there's more than one.
Yeah. Well, just to make sure that we're literally all on the same page, because we're flying blind here.
I know. Yeah, this is the right slide. So this is probably the slide with the most words on it. And again, I apologize for all of these words, but there really is a lot of research taking place in the 5 weeks that we're here. So what this slide says in a short and sweet version is that we're using a combination of science techniques to determine the types of iron-binding compounds that may help diatoms. What is in the waters here in the Southern Ocean that may allow diatoms to acquire iron that they may not be able to get themselves? And so that's really the basis of the research here, and then there are lots of different components of that research, whether it be biological or chemical, in order to help answer those research questions. Next slide, please.
Okay.
Okay. I thought I lost you on the phone, too. I was going to panic. All right. So how do we answer these research questions? Well, you need to collect the organisms in the water that you're trying to learn more about. And so on board, we have two different collection methods that we've been using over the past 4.5 weeks. And they are referred to as rosettes. They're basically large frames that hold something called a Niskin bottle. So in the picture you're seeing here, the main picture on the slide, the long gray bottles are the Niskin bottles. And they can be automatically closed, or they can be computer closed via a long cable. You just click a mouse and the bottles will close in order to collect samples at various depths. The one that you're looking at here in this picture is the trace metal rosette. There's some special precautions taken in order to make sure that the water samples from this rosette do not have contamination from the iron that may come from the ship, that may come from clothing or tools or other things that are on board. And so there are gloves worn and sleeves worn and certain procedures that are done to make sure that when the water comes out of the bottles, it has not been contaminated by iron. Because we're looking at such small levels of iron here in the Southern Ocean, any type of contamination, even dust, could set off the data from what the actual numbers are here in this region. So I have a video, and we'll see if this will work, and hopefully we don't bump everyone off, but this is a little 1-minute video showing the trace metal rosette actually going into the water.
Okay, so we're getting the video started. And if you remember the video, you can kind of explain what is going on. It's about to drop. It's like over the edge of the ship right now.
Okay, so that's the starboard A-frame, and the gentleman in the picture on the left is Rich Thompson. He's one of the marine technicians on board, and the woman on the right is Holly Martin, and she is another marine technician, and they are responsible for deploying the scientific equipment. And so that A-frame, as it moves outward, the winch is used in order to lower the rosette into the water and then drop it down to predetermined depth, sometimes as far down as the bottom, depending on the request of the scientist.
All right. And we just ended the video. We're going to go back to your presentation real quick.
Okay.
Are there questions from the chat related to anything? We're not seeing the chat screen ourselves right now, so if there's any questions that are related to what Cara or just covered, this might be a good time to ask a couple of those if they're related to the CTD, and then we'll move forward.
All right. Yeah, there were some people that the bandwidth of the video did impact them, so we're just getting some feedback about that. But I think some people were able to see it too, so it's a little bit of everything. Again, we apologize. We're just testing that out to see if it worked or not. I don't see any questions at the moment. So the last picture that we were on was the CTD and the Niskin bottles.
Mm-hmm. Okay, so we can go to the next slide if you'd like.
Okay. We're there. We got conventional CTD sampling.
That is correct, yes. This is the second rosette that's on board, and this rosette holds 24 bottles, and each of those Niskin bottles can hold 12 liters of water, just to give you an idea of how much we're collecting with each cast of the CTD. This one is not a trace metal clean rosette, and so this water that's sampled and collected is usually used for the molecular biology and some of the biological sampling. Next slide, please.
Okay.
Both of these rosettes are equipped with a CTD, which measures conductivity or a measurement of the salinity or saltiness of the ocean, the temperature, and the depth. And we can also add other sensors onto the CTD to test for oxygen or for light transmission through the water. And one of the nice things about having the CTD is that you can get a real-time image of what's happening in the water column, and that's what you're seeing there on the right of your screen. So Barry is one of our ETs on board, our electronics technicians, and he runs the CTD, and he'll fire the bottles to collect the samples, and he'll help in sort of interpreting the CTD screen for us so that we can, or not we, but the science team can make decisions on where to collect water samples. So all of these pieces of equipment are useful in helping to actually bring the water on board. And once it's on board, there's a couple of different things that happen. And one of the main objectives of this cruise was to run some incubation experiments. So next slide.
Yes, we're changed, and you're getting feedback. I'm muting you. Okay.
Yep. Okay, thank you. Okay, are we on the incubation experiment slide?
Yes.
Okay, so this is one of the setups for one of the incubation experiments. If you've been reading my journals, you've probably heard a lot about these incubation bottles. And so what happens is the water that comes out of the ocean is put into 50-liter carboys, which you can see there in the center of the slide. And these carboys are used to distribute water into small incubation bottles. So the incubation bottle is the little white bottle that you see there near the bottom of the slide. The incubation bottles are then set up and spiked. And what we mean by spiked is that there are different nutrients or different groups of nutrients that are added to each bottle. And those are represented by the 6 different colors that you see there next to the incubation bottle. The nutrient combinations or levels are determined by the science team, and that's something that's part of their actual hypothesis, and so that's not something that's going to be shared with you until they publish their information. Once they have all of the bottles set up, we're going to transfer these bottles to the incubation. So Jana, if you want to go to the next slide.
Yeah, and there's a question by Lucas who wants to know, how do you differentiate between contamination and real data? And you can take that whenever you want to.
Let me see if Travis is on here yet. No,
Yep,
of course.
Is
he?
Yep, he just typed in.
Oh, there you go. Thanks, Travis. Travis is a trace metal guru, so I'm going to let him field that one because, Lucas, as you might know, chemistry is not my forte, so so we'll let that conversation keep going. So we're on the incubation van. So the bottles are taken to 2 different locations. One is an incubation van that's in the hold of the ship. And that's the top left-hand picture. And the bottom left-hand is showing the inside of the incubator, the bright blue light that you see there. It's a place where light is provided for the water for 24 hours, and it's also kept at a constant temperature. So they're really incubating, kind of like chicken eggs would incubate in warm temperatures, whereas these diatoms, because they live in cold waters in the Southern Ocean, are incubating in colder temperatures.
I like to think of them as my babies incubating at very cold temperatures.
We also do the same for other samples that are actually stored outside, and this is a really interesting piece of equipment. They're on-deck flow-through incubators, so the actual water from the ocean is being pumped through them to maintain a temperature that's ambient or natural for the environment, and also they're getting the same light that they would get if they were in the water because they're outside. And so when the sun sets on the ocean, the sun is setting on those incubators. I think I can actually do it this time. There we go. So randomly, well, not randomly, every few days we take samples out of these incubation bottles, and the samples are distributed to the chemical researchers and to the biological researchers. And you can see some of the different analyses or preparations that are done on these samples. And what I want to emphasize is the arrow that's in the center. The reason it's pointing to both sides is because even though these samples might be distributed to the chemists or the biologists, the information is being used collectively. And this truly is a collaborative project. To hear all the scientists talk together about how the chemistry is influencing the biology is very interesting to me. And so they're working together to make sure that whatever data they collect is coming together in order to help tell a story about not just the biology, but the chemistry of the Southern Ocean. So before I wrap up, I just want to show you some images of some of that sampling. Again, this is not all of it. But the first is the trace metal sampling, which takes place in that plastic bubble in the lab. This is sort of an unknown location to me because not a lot of people go into that bubble, again, to limit contamination. But you can see a picture down there of the inside of the bubble. It has lots of filters that are creating positive pressure, blowing air out in order to remove contaminants. And trace metal clean techniques are used inside of that bubble to limit contaminants as well. So there are a lot of questions that the trace metal team is asking, looking at different concentrations and the chemical forms of iron, and again, looking at how water chemistry influences biology, and then vice versa. Also part of that chemistry is the nutrient sampling. This is Will Abbott, who spends a lot of time in the wet lab, and he basically works very odd hours whenever samples are available to him, testing for concentrations of things like nitrates and silicates. As I mentioned before, diatoms use silica to create those frustrals, or those little houses. So where you find silicate, you probably, or you may find levels of diatoms because they can use that silicate along with these other nutrients to grow and possibly use their chemical reactions. There's sampling done in order to run DNA and RNA sampling in the future. So in the far left-hand corner, the picture that you see there is Kim Powell. She's from Old Dominion University, and she's using a pump that's going to draw water out of the collected carboys and run it through filters. And the filter is the center picture that you see there. On the far right-hand side, you can see what happens after maybe 20 liters of water is run through a filter. The red circle is the holder of the filter, and inside the filter, normally that filter would be clear, but you might notice that it's a greenish tan color. That is all the biomass or biological material that's being collected on that clear filter, and it starts to basically show color because of the chlorophyll that's inside of these different microorganisms. Those filters are flash frozen and stored, and they'll go back to the Old Dominion University and the University of Rhode Island labs for further analysis on DNA and RNA to look at the genetic makeup of these organisms as well as any genetic changes that may happen. There's a flow cam on board that helps us actually get real-time images of some of the diatoms that are in the samples. So the picture on the left is showing the computer screen that actually shows the diatoms as they run through this tiny little tube. There's a camera and a magnifying lens that's going to show you the different sizes of the diatoms. And so we can actually record photographs of the microorganisms. You can look at those within a given volume of liquid to compare from one day to the next to see if your samples are growing. And you can also use it to look at the actual different species of diatoms that you might find in your samples. And then there's some cell isolations and culturing that are going on, and I have to say before I explain just what's going on that when I explain this, note that we're on a moving ship, and especially Laura, who is in the top right-hand side from the University of Rhode Island, the day that I took that picture, we were rolling around a lot, and I was very impressed by her ability to isolate such small cells. So Bethany is in the other picture, and they're both looking at water samples and actually using pipettes to take out individual diatoms and then move them to isolation units so that they can then be cultured or grown. And one of the reasons for this is to be able to take samples back to the lab of actual diatoms from this region. So instead of ordering samples of diatoms that might not come from this region, you can actually grow your own Southern Ocean diatoms for future experiments that I think is pretty cool that you can then have those available. But it's not all about the science, and I apologize for the blinding orange, but most of the safety equipment that we wear is orange, so this slide is the orange slide. We have a great group of people on the ship, from the support staff to the science team, and we're having a really good time getting to know each other, doing crosswords together. We've had a barbecue. There are card games going on at night, movies, foosball table, and ping pong tournaments. So we're really having a wonderful time getting to know each other, and it's amazing to see all three science teams sort of coming together and working so well and working so hard to accomplish a goal. And we've also, I can't leave this out because this is one of the highlights as well. I've learned a lot on this trip, and I've also seen a lot. And the wildlife list has grown since I had to send this PowerPoint in last week. You can see this is the white slide because everything's white except for the animals. There's a lot of ice here, but we're seeing lots of different species of birds. We've seen a couple of different types of penguins, different types of seals, and one of the highlights was the orca or killer whale sightings that we had just the other day. And finally, just so you know, this is not the end of things. Once we get off the ship on the 14th or 15th, this is not the end of the research. So we're doing a lot of sampling, we're doing a lot of collecting, and there are a lot of samples that are in a freezer, in fact 2 freezers right now, and those freezers are overflowing. We're looking for new places to put samples to be able to ship them back to the different universities. There is going to be a lot that happens to these samples over the course of the next few months in order to learn more about the samples that were collected. And there will be a lot of data analysis and a lot of publications that come from this. On my end of it, I hope to do a lot of education and community outreach. I have lots of ideas for lesson plans. I'm giving some local presentations in the Quincy area, and there's even talk between Bethany and I about possibly being able to utilize our close distance to the University of Rhode Island to maybe bring some students to learn more about some of the molecular techniques that are taking place there. So it's not just a we're over and done. As soon as this trip is over, I think Bethany and I, along with the other researchers, have established a really good relationship, and I hope to see that continue as we move forward. So that's the end of my presentation. There are lots of people certainly to thank, and I think you can read through that list. That's some of the research, certainly not all that's going on here. But if you have more questions, you can certainly read my journals. And you can also, I think, we'll be ready to take some questions soon.
Yeah, great presentation.
We're taking a virtual applause.
Yeah, virtual applause. We can do that. We can do that. It's you set your status and you look under there and we can applause for you. Yeah, so great presentation, nice balance of science and everything. Let's see, there were some questions. Lucas has been asking a lot, and Travis, thank you for responding. Oh, yay, and we get to see your face, too. So let's kind of back up because there might be some people that didn't get to read the chat as it was going on, and they might be just joining by phone. And feel free to re-answer some of these. We want to know how many species of diatoms are there, and have you found any new ones?
Ooh, that's a really good question. So there's estimated to be about, and this is really approximate because it's a very species-rich group of organisms. So globally, there's thought to be over 200,000 species of diatoms. There's fewer than that in Antarctica, but we've seen, in the orders of, in the region we've sampled, I would say we've seen at least over 100 species. And so that's where the FlowCam gets to be really helpful because we have a video documentation of what we're looking at. And classic phytoplankton ecologists used to do that work under microscopes too, which is really difficult to sit there and count individual species. So the photographic record will help us enumerate those when we get home.
Yeah, I can't imagine counting all of those. That's impressive. Let's see, there was another question that you answered already, Bethany, but I think it was also a good one. It says you are sampling a bunch of sites. If you find something really interesting, are you able to go back to the site, or is it one and done for this trip?
Well, a lot of that depends on our time and where we are. And so because we knew some sites would be interesting, we've set up these incubation experiments. We plan to go back to some of those sites so that the site where we set up the first incubation, we were able to go back 2 weeks later and already had seen signs of spring, slightly higher. So the days are getting longer, and that's really noticeable with the light coming into my cabin at about 5:30 in the morning. It was dark when I was waking up at the beginning of the cruise. So we see that shift, and the phytoplankton, because they're photosynthetic, are responding to that shift as well and growing in response. So that longer light period allows them to start dividing a little more quickly, and we saw signs of that when we went back 2 weeks later. Other sites, I think probably the captain of this vessel, if I told him we wanted to go back through the ice because I forgot something, that might not be a very popular decision right now. So it depends on timing and in this thick ice, how much progress we can make.
Yeah, I guess related to that, are you going to be coming back to this area in the future, not this year or in a couple of years, or how long does this particular research project run?
So I'm laughing because it will require me to go back to the National Science Foundation and ask them for put another proposal in to request more funding. So this particular proposal is funding this research, has one trip planned, and then we're bringing a lot home with us. So these diatom cultures, experiments will continue in the lab, and the collaboration will continue past this cruise in kind of a lab-based setting. So this is our one shot at a field season as part of this collaboration.
All right. Before Joanna Spring and some students sign off, they wanted to say thank you for this opportunity. Apparently they're writing some college essays and your students miss you. So
You want to say goodbye?
you can
Yeah,
say goodbye.
Good luck on your essays, and I'll see you in, I think it's something like 3 weeks, so I'll see you soon.
Awesome. Okay, thanks for joining us there. Some questions? Wow, that was awesome. Seth Connors would
Thank
like
you.
to know, Seth Connor is asking, the lamps in your incubation areas look like they are fluorescent. Do you have any concerns over differences in the frequencies of light and how they might affect the growth of your organisms?
That's a great question. So they look really fluorescent, and they're actually blue. And you can imagine if you're under the ocean, a lot of you guys have snorkeled and been swimming, it's really blue there because of the way the ocean absorbs light. So these lights are actually not mimicking the light right on top of the ocean. They're trying to mimic the light, the phytoplankton in the spectrum, the phytoplankton see under the water. So it's a very cool setup, and it's we've got a researcher that's got bright orange foul weather gear, and so he glows in the dark when he goes into the van, which is pretty awesome.
Okay, let's see. Lucas says, you talked about using ocean water to incubate. How much of that goes back into the ecosystem, a.k.a. what is the ecological footprint of the ship?
He's an environmental guy.
Yeah.
Well, I can say that anything that has been touched by chemicals or by any production that happens in the lab, there is an extensive protocol for waste that is actually stored on the ship and then will return to port with us. Any water that is returned is water that has just had the biomass removed from it. There's also, I've been learning a lot about the Antarctic Treaty, and there's a lot of, I saw Lucas ask another question about invasive species, so I'll try to tie this together. The Antarctic Treaty has, and I actually read some of the, basically the guidelines and the bylaws, lots of requirements for how to store waste, and all of the waste that comes from Antarctic bases and Antarctic ships is processed back on lands like South America or in the U.S. So none of that is actually taken care of or left here in Antarctica, and that is also the place where you'll find the information on invasive species. No invasive species are permitted to come to Antarctica without a special permit, and my understanding is it can be difficult. And I'm still working out the ship if it's carrying invasives on the bottom. I'm still working on that question to see if I can find out the answer.
Okay, great. Yeah, you answered the question that I was going to do next. So Guy wants to know, where is Harambe?
Harambe? I don't know what that is.
Maybe that was somewhere. So Guy, if you, bogus question, maybe. You're just trying to throw us off. We don't know where it is either. Okay, well, if Guy would like to comment on that question, we would like to answer it. Okay, let's see, what else? I think there were some people that signed off while you were there. The volleyball team was signing off, Turtle in New Mexico, and Karen. So anyway, they were all wishing you a safe trip, and thanks for presenting. Looking to see if there's any other questions I missed along the way. So while we're waiting, where does your cruise take you from here? What's the next little bit?
Good question.
Well, we're going to plow through some more ice. We're trying to, we're going across a very steep shelf along the Antarctic Peninsula that also overlaps with this long-term ecological research grid. And so the depths very close to shore are very shallow, and then as we move offshore, the water gets very deep, and the chemistry changes along those gradients. So we've been Kristen Buck's team in particular has been mapping those gradients going back and forth across the shelf to look at things like nutrient dynamics and gradients of the metals that are available for the phytoplankton. And we'll be correlating that with the phytoplankton community composition that we see, both using the flow cam as well as genetic measures back in the lab. So we're all kind of collectively sampling that. We had hoped to push south of where we are, and I think the ice is probably going to shut us out pretty quickly from doing that. So the next set of plans is to go back offshore into that lovely Drake Passage and collect a little bit more water there where it's free of ice, but maybe not free of wind. And we're going to be testing some new equipment for future field efforts.
All right. Kip wants to know, what will be your first meal when you get back?
First let me say that you really want for nothing on this particular cruise. We have some incredible staff in the galley, Mike, Ralph, and Andy, and between the baked goods and the meals, we had a big Mexican lunch today. We had Italian day yesterday, so I really want for nothing, to be quite honest. The only thing, and I'm sure this will make some of the staff laugh because they know me so well, is I'm ready for a big, giant fountain Diet Coke. So that's what I'm going to be looking forward to.
As senior leadership on a vessel, we're privy to the office that Cara and I are sitting in has a Pasquini espresso maker behind us, and the crew lovingly keeps this thing in good working order. So for those of you know how much I need my coffee to function, I do get a daily latte, so that's a huge treat. But there's a Chilean sandwich place called Lomitos in Punta Arenas, and they have big sandwiches and big glasses of icy beverages. So I think a lot of us are going to look forward to having lunch there when we get back to port.
That sounds lovely, all right. So Nicole has questions from the class. Has everyone on the ship had some time for themselves? And then the second question, is the iron in the water the same as in the Arctic? The same in the Arctic as in the Antarctic?
Well, I can field the first question. Hi, Mrs. Kamissis. I would say that everyone has definitely had some time for themselves, some more than others. But we have had some days where, like today, we had a wake up for a 7 a.m. cast, and we were stuck in some ice. So there are always those moments where you get some time for yourself. And as I said, most of us have our own rooms, so there's always that time in the evening when you're getting ready for bed where you can have that time for yourself. But otherwise, everybody is working really hard here, so it's great to see that sort of commitment to the science. The iron question, I don't know, either Travis can answer or Bethany, do you know?
Yeah, I think I can take a good attempt at answering this. I don't necessarily know the range of iron concentrations that Travis might know, but the Antarctic, if you think of the map of the world, the tiny, tiny tips of southern continents, there's just a lot of water moving around Antarctica without a lot of land to get in the way. And the ocean around here is pretty deep, so most of the iron comes into the ocean from land-based sources. The Arctic's really different because there's more land, much more shallow shelves, so you get sediment-sourced iron supply up in the Arctic Ocean that you don't really get down here. So that's one difference. So the Arctic is less severely iron-limited than the Southern Ocean. I should answer my dad's question since I see you here, Dad. Bill Jenkins has a question about robotic collection equipment. One of the principal investigators that runs the LTR sampling program is also an expert with some ocean robotics. So they do deploy those types of things off this vessel. I personally don't have as much experience working with remotely operated vehicles and things, but this ship certainly is set up for it. And the other day I was down in the electronic technician office, and they were working on some remotely operated underwater camera systems that look really neat and fancy.
Okay, great. Let's see, there was another question. What is the combined cost of manufacturing, transporting, and deploying a rosette complete with Niskin bottles? From Lucas.
I don't even know. It's something we don't want to lose. Occasionally the ocean will exercise its will and engulf one of these sampling arrays, which is not what we want to see. The trace metal rosette is on a Kevlar line. It's about 3,000 meters of special Kevlar. So even the wire that it's on that also has data conducting capabilities, I'm guessing is pretty darn expensive. I just don't know the numbers behind it. And the bottles that you see on the rosette in the picture that was put up, those we're borrowing from a researcher at Rutgers. So trace metal chemists are a very finicky group of people, and they tend to own their own sampling equipment because they don't trust anyone else. So the U.S. Antarctic program owns the epoxy-covered cage and the Kevlar line, and the bottles come from researchers. So they ship these bottles all over the world because they know how they've been handled.
Okay, we have some questions coming from your school. There's a bunch of students in the auditorium there. So far we have from Amanada, sorry if I mispronounced your name. How far has your research gotten?
I'm not sure if you mean in terms of the experiments or in terms of distance. I don't have a total distance yet. I'm waiting till the end of the cruise to find out how many miles we covered over the course of these 5.5 weeks. In terms of the research and the incubations that are being conducted, the first incubation experiment is finished. That was 12 days? 14 days? 14 days. And we're currently in the middle of the second and third incubations. There are other research experiments that are going on, some individual projects by some of the graduate students, as well as ongoing sampling at those LTER grid lines. So we're still researching and sampling up until, I think, next Tuesday is when we start to depart.
Yeah, we'll try to, we need to pack up all this equipment, and it's nice to be packed up before we cross the Drake again and start really banging around. So we'll try to get most of the sampling equipment packed before we transit the Drake, which will probably be sometime on the evening of October 10th.
And it'll take, I think it's at least 2 days to cross the Drake, another day to travel up the coast of Argentina and through the Straits of Magellan. So that has to be taken in account as well.
Okay, and another question from Brianna. What's the most interesting thing so far?
Well, you know, the sightseer in me wants to say the wildlife and the ice, but I honestly have to say that, and my colleagues can attest to this, I usually am the anti-chemistry person. In fact, when I first heard about all the chemistry that was going to be on this research cruise, I panicked a little because I really didn't think that it was going to be something that I was going to be able to understand or was going to find interesting. But that's actually not true at all. The research team is so helpful. They put up with all of my questions. And I have learned such an enormous amount about how the chemistry can interact with the biology in an interesting way and in a way that I think can help me be more excited about that component of my teaching. So for me, I'd say, you know, in addition to the really cool stuff we're seeing, you know, just as we transit around, the science is actually really interesting, and I'm very excited to be able to say that.
All right. From a new teacher there, Patrick.
Well, the orcas were pretty cool too. We've had wind. We've had a ton of great days. So the days that the sun has been out have been few and far between. So we had this evening that was sort of free of work, and a lot of us went up to the bridge, and then these orcas came, and it was like this perfect sunny night with a beautiful sunset. And the mate on watch, oddly enough, used to be a whale watching captain. So we took the Palmer. The whales were close to us, and he knew how to drive to not disturb them. So they stayed near us, which was really amazing. And so people got quite a show from these whales with a setting sun in the background. So, you know, in addition to that kind of science, the wildlife can still blow you away.
Nice. Okay, we have also from your school there, a new teacher, Patrick, wants to know what are the safety features of the ship?
Oh, wow. Well, first of all, hi, Patrick. Welcome to North Quincy. I look forward to meeting you. There's a lot of different features on the ship, just in terms of the crew that is there to make sure that you're being safe. There are flotation devices, flotation jackets, flotation Mustang suits, safety harnesses, your firefighting equipment, I mean, hard hats like we had on at the beginning of the PolarConnect event. We have steel-toed boots that everyone is issued. And then just for the cold, prior to getting on the ship, the U.S. Antarctic program issues everyone heavy-duty winter gear, so big puffy jackets that are called Big Reds, lots of fleece, lots of waterproof gear, things of that nature. And the crew on board is extremely professional and very aware of safety procedures that are needed for each piece of equipment. So on board the Palmer, the scientists aren't actually putting out the CTDs, and so there are well-trained folks who are doing that for us.
Okay, another question from there. It's from Bev. Are you bringing home samples to your school?
I don't know. I haven't asked that question yet. There is discussion. Bethany and I have discussed maybe using samples that are local to the New England area, but I don't know. Maybe if I ask nicely, I can get a little sample of something.
Yeah, and if North Quincy wants to name and adopt Diatom pets, we can certainly do that.
Absolutely. Oh, there you go,
Bev. Jackson would like to know, how are you adapting to the close quarters on the ship?
I think Bethany can attest to the fact that I've smiled every single day that I've been on this boat. Even I was pretty down and out a few days with a pretty bad sinus infection, and even so, I'm enjoying every second of it. I don't mind it at all, and for me, this is I kind of feel like it's second nature to me. So I'm really having a good time.
Okay. There are some other people that are complimenting you about things. Oh, somebody asked, how cold is it? Mickey.
We had a number of days where we were hitting winds between 50 and 70 knots. So you go outside and wind like that, and you know, even if the real temperature is around freezing, those winds are extremely cold. Oddly enough, in the ice that we've been in, and that was in open water, but in the ice that we've been in, it's been snowing, and it feels balmy. You know, it's like that 30-degree Fahrenheit snowstorm with thick, heavy, kind of wet snow, and it just has felt warm and pleasant the last couple days.
Absolutely.
T-shirt weather in Antarctica.
Okay. Let's see. Now the questions are getting silly from a few of your students there. Anyway, we'll go to Lucas's question, and then we're going to start wrapping this up. So if you have any more last-minute questions, make sure you put them in the chat. But Lucas's question is, will rising levels of atmospheric CO2 and ocean acidification result in larger, smaller populations of diatoms in the Southern Ocean? If so, then what will this mean for the rest of the ecosystem? Nice.
I'm going to let you take that one.
Okay, she's passing to me. Well, I think, Lucas, this is a great question because it really gets into some of the biology that we're studying. And I think down here it's actually the iron that's going to control the system. So in some ecosystems, you pump more CO2 in, and that's actually like a good thing for phytoplankton in terms of they're able to respond and grow. What I think is going to happen in the future is one of the things that oceanographers have a challenge to predict is how the iron cycle may change as the ocean gets warmer and the pH starts to decline. And so if iron concentrations in the Southern Ocean drop even further, that's going to present a real challenge for some of the diatom biology. So I would say that that's still an area of active investigation because of a lot of the control on growth is coming from those limiting iron concentrations.
And just to let Mohammed know, no, I cannot bring back a penguin. So I can't bring back any animals from Antarctica, even though I would like to.
Although our marine programs coordinator's 4-year-old daughter will be a penguin for Halloween, so we challenge you for that penguin Halloween costume this year.
And sorry about that if I switched screens on people. I knew people were logging off, so I wanted to get them to the polling area and say thank you for joining. And Lucas has one more question, so we'll take that. What specifically do trace elements come into play in diatom photosynthesis?
All right, I think Travis put it in script there, but yeah, they're definitely part of various chains in the process. So the overall reaction of photosynthesis, it looks like it's just one step, and I know that many people know that there's a lot of steps in the middle, and those trace elements come into play, helping enzymes do their job and also helping with electron transport, things of that nature.
All right, and your school is getting ready to leave you, so I think you might want to say goodbye to them.
Bye, guys. Have a great day. Thanks for joining me.
Yes, very good, Igor, joining us. All right, they were saying goodbye there. Yeah, thank you. It was good. Let me stop the recording.